It's 2:30. Hello, everyone. Welcome. Erin, will you be letting people in from the waiting room? Wonderful. Thank you so much. My name is Ann Price. I'm a volunteer for Angie. I work for New Jersey Audubon. Thank you so much for joining us today. With me, we also have Erin Rounds. She's the Angie secretary, and she's co-hosting this program today. We're excited that you joined us for today's workshop, Muddy Jeans, as part of Angie's 37th annual winter conference. I really tried to count up how many I've been to. There's a lot. A special thank you to our conference sponsors, PSE&G, New Jersey Natural Gas, and our anonymous donor who's a friend of science and environmental education in New Jersey, and to BASF and Grounds for Sculpture for supporting our field experiences. Also, we're placing the link to our YouTube channel in the chat room where you can see all of our recorded content from past conferences and forums and other sponsored events. Wow, Erin, that was like you had it set and ready to go. Wow. Check out the YouTube channel. We hope you subscribe. So there's plenty to see there. Just a few reminders for the session. We ask that everyone keeps themselves on mute throughout the presentation. Yeah, I'm supposed to have that slide up there, sorry. We ask that you have it on mute throughout the presentation. If you do have any questions or comments, please put them in the chat room. There will be time for live questions toward the end of the presentation, which you can either raise your hand or use the hand raising icon and we'll let you ask your questions. So let me share this slide with you. Did I put it up correctly? Am I doing okay?
Yeah, looks great.
Thanks. Okay. So today's workshop, Caitlin Gannon from Jacques Cousteau National Estuarine Research Reserve is our presenter today. What does environmental DNA, sea level rise, and mosquitoes have in common? In 2016, the Jacques Cousteau National Estuarine Research Reserve, Rutgers University, and coastal county mosquito control agencies teamed up to begin a research project which examined the impact of rising sea levels on salt marsh mosquito populations using methods such as soil core sampling and eDNA analysis. This locally relevant research project inspired the creation of teaching modules which can be used in the classroom. In this presentation, we'll review Muddy Jeans, an activity from the teaching modules, which simulates how scientists obtain and analyze eDNA and how this information can be used to make informed decisions about species management. Caitlin, please introduce yourself and let's get started.
All right, well, thank you so much. Thanks for having me. And thanks for joining my presentation today. I will start sharing my screen. So 1 second. Screen one, presentation, slideshow. Okay, does that look good?
All good, thank you.
Okay, great. Well, thanks again. Again, my name is Caitlin Gannon. I'm the education coordinator at the Jacques Cousteau National Estuarine Research Reserve, or JC-NER as we like to call it. We are an organization that's managed by Rutgers, but we are part of the National Estuarine Research Reserve Network, which is a federal operation or group that's gets NOAA funds, and then we all are managed on some sort of state level around the country. So there's actually about 30 reserves in total throughout the United States, and the one in New Jersey is managed by Rutgers. So I was very fortunate to be part of a research project starting in 2016 that involved a lot of Rutgers University staff and faculty, as well as county mosquito control agencies. And the title of this project is called Understanding the Connectedness of Climate Change, Salt Marsh Resilience, and Nuisance Mosquitoes. Along with the science, I was brought on board to create K through 12 activities related to the work that was going on with this scientific research, as well as other outreach and engagement products, which I'll show you a little bit later. Feel free to put questions in the chat. I'll ask you guys some questions as we go as well, to kind of test your knowledge as we go. And yeah, it'll be a lot of fun. Feel free to have any questions throughout. So hold on, I can't, there we go. So my plan for today is to provide some background information related to the research that was going on. If I jumped into the research project right away, it might be a little confusing. So I just wanted to provide some background information about sea level rise, eDNA, mosquitoes, how it all came together, all connected. I'd like to also do just a really short demo about what the activity is all about. And then I'll review the whole activity with you and then show you where you can find these activities and other resources that could help when you're teaching. So just to start things off, you know, it all really starts at how our coast is changing. I think a lot of us are observing more frequent flooding, even on a sunny day. This picture here was just right after a nor'easter, I think, that hit last year. And this is on Great Bay Boulevard where I work. I work at one end of this road. And then at the other end of the road is the Rutgers Marine Field Station, which sometimes it's really, really difficult to get to when we have a storm that just came through, or if it's a king tide, it's often flooded. So it's very powerful, you know, it's sometimes overwhelming to think about, but these are things that we do have to consider, you know, if we want to adapt and make changes and plan when it comes to changes along our coast. There's a lot to consider. But, you know, in terms of sea level rise in general, I know most of you probably understand the concepts of what's going on, but just to do a quick recap, you know, a lot of the rising water levels are occurring due to land ice that's melting, but also a lot of it has to do with absorption of heat in the ocean. And as a result, it's creating thermal expansion. If you've done the experiment with your students before of heating up a you know, a bowl or container of water, and then you put the straw at the top and then let the water, you see over time, and as the water heats up, the water goes into the straw and travels upward. You know, you've probably understand or know what I'm talking about. I wanted to share though with you today, there's a really neat, and I know it's been difficult for teachers over the last year or so, so I'm trying to give you guys as many online simulations as I possibly can. So here's one that's really cool. I'm just going to interrupt my own PowerPoint for a second with something else, if you can still see this. But this is an online simulation of that exact phenomenon, right? So we're heating up water, and with more heat and over time, the water molecules inside are going to get more busy, start moving around more, spread apart. And then as the temperature heats up, you can see water is slowly going up this little tube right here. And I can put this link in the chat for you guys at the end, or I'm not sure, maybe it'll get emailed to you later, but definitely a really great simulation from the Lawrence Hall of Science. So I just wanted to share that real fast. Pause that. So thermal expansion, big contributor to sea level rise. It's likely that 2010 to about 2050, sea level rise in New Jersey will increase by 0.2 to 0.5 inches per year. which doesn't sound like a lot, but if you put it into perspective, that's 2 to 5 inches per decade. The global, also known as eustatic average, sea level rise is occurring at 0.13 inches per year. Now, why is it higher in New Jersey? If the global average is 0.1, but New Jersey it's 0.2 to 0.5. Does anyone have an idea of, maybe they can put it in the chat of why they think it might be more in New Jersey relative to the rest of the globe? Not all sea level rise is created equal. All right, densely populated, elevation, development, coastline. Do another few seconds for some answers. Shape of the continental shelf, type of coast. You guys are on the right track, definitely. So one reason is a phenomenon called glacial isostatic adjustment. So if you think of the tectonic plates that are all over the planet, and the tectonic plate that North America sits on, there are really, really heavy ice sheets a long time ago near the northern end of that tectonic plate. As a result, it was pushing down on the land and kind of creating a bulge, it pushing the land up. And then over time, as ice mass melts and land ice especially melts, that sort of has a rebound effect. So the ice sheet will decrease and you have this seesaw effect of the land kind of coming back down. And so sea level rises relative to the land because the land is actually going down. Another reason is the area or the coast that we live at. We have a lot of development on barrier islands, and barrier islands are not very stable. It's a lot of sand, a lot of sediment that moves around because of the movement of the water. So the land along the coast is sinking just because we are on very unstable ground, but also we're withdrawing a lot of water from underground aquifers and not replenishing it at the rate that it's being taken out. So the land is kind of going down as a result of both of those things. If you think about, going back to how this sediment is very dynamic and just moves around a lot. If you think about another coastal city, like, for example, New York City, that's built on bedrock. So it's much more solid than a barrier island and probably doesn't move as much as the New Jersey shore. So these are all factors that go into why New Jersey especially has much more dramatic or more variability when it comes to sea level rise. So now, how is sea level rise impacting our salt marshes? Here is a map that I got from New Jersey Flood Mapper. If you've ever used that website, it's a really great website and shows you different simulations of what would happen if sea level rise were to occur in different scenarios. But for right now, this is just a basic map of the Mosquito River meeting Great Bay. Great Bay is down here. And then the Atlantic Ocean is somewhere over here. We have Atlantic City down here. Just to give you some perspective, this is the township of Galloway. And you can see these salt marshes all throughout the edge of the Mosquito River. So we have some on the south end, we have some on the north end up here, and then up here is pine barrens mostly, right? So I made a map in Flood Mapper of a scenario that shows what would happen to our salt marshes with 1 foot of sea level rise. So to explain the map real quick, we're looking at salt marsh wetlands, which are in green. And then the pink part is where salt marshes are expected to move or retreat as a response to sea level rise. So salt marshes and habitats will change as these other factors change, right? So salt marshes are likely to move or retreat back as sea level rise increases. And that's what all this pink shows. This is projected areas to where those are going to be new areas of salt marsh eventually. Same thing with the dark purple. but the dark purple usually is, I think it means that there's an impediment, like a roadway passes through here. So it might flood this road and then create new salt marsh maybe on this side. So what would happen with 2 feet of sea level rise? Our marshes change even more. So we still have our green, those are all salt marsh. And then our area of expected retreat grows. So new areas of salt marsh are this pink. But then also we have this yellow, and this yellow means that this was at one point marsh. Now it's just open mud flat. So it's just mud, water, no vegetation really. So that's a huge dramatic change with just, 1 foot of rise. So marshes are definitely going to change quite a bit, even between 1 to 2 feet of sea level. So knowing that marshes are going to change as sea level rise changes as well, the big question for the researchers in this project was, with changes to the salt marshes that we know are occurring, how will this impact mosquito species that breed in the salt marsh? Not many people know, but there's about 4 to 5, at least in New Jersey, species of mosquitoes that prefer to breed and lay eggs in salt marsh habitat. And local county mosquito control agencies treat salt marshes with a larvicide to combat this. So coastal communities can have, areas where they're not being impacted by nuisance mosquitoes. And mosquitoes are a nuisance, but this is also important because they spread disease. So we want to make sure that, those populations along the coast that breed in the salt marshes are managed effectively. But with the changes in the salt marshes, because of sea level rise, how is this going to impact breeding mosquitoes for effective management in the future? They're really good at finding mosquito hotspots now, but what are those hotspots going to be looking like, or where are we going to find them later on in the future as our salt marshes change? So to answer this, we need to find the mosquitoes. And sometimes that's actually not an easy thing to do. You can go out into the salt marsh and wait until you get bit by a mosquito, but that's not very effective. So how do you find where, not just where mosquitoes are, but where are they laying eggs? Where are they breeding the most right now? And to do this, the team came up with finding mosquito DNA. So going out into the salt marsh, collecting samples, such as soil samples, water samples, and we collect them in various different ways. One of them is to use a soil core like this and literally just push it down into the ground, get a sample of soil. Another way is to analyze salt marsh pools and the water within different pools and ponds in the marsh. And some of those areas are really, really difficult to access. So we've used drones to help us with that. This drone right here would have this contraption at the bottom of it, and it would sort of dip it into the marsh and go up and collect a sample of water. And these samples, whether it was a water sample or a soil sample, they're filtered, and that's to make sure that the DNA inside of the sample was isolated and it can be put through a machine, and then the machine reads what DNA is present in that sample. So really, really interesting. Before I go more into eDNA, I just want to show you a video. You might have heard of eDNA before. It's like a fairly new method of surveying what kind of species are present or absent in a habitat, which is kind of neat. So I'm going to play this for you right now. I hope, actually, I don't know if I'm sharing sound. So 1 second. Here we go. Okay.
Imagine if, from just a drop of water or a speck of dust, we could detect any living thing. We now can thanks to environmental DNA, or eDNA for short. All creatures leave traces of DNA in their surrounding environment, via skin cells, hair, scales, anything really. And this is where the magic happens. Scientists have worked out how to extract this DNA from the environment and use it to identify the animal, plant or other organism that left it behind. We start in the field, collecting samples such as water from rivers, lakes, estuaries, dams, you name it. eDNA can also be found in terrestrial samples like soil. Next, the lab coat goes on and eDNA is extracted. The unique sequence of each DNA molecule is analysed for matches against a database of species. Once we get a single or multiple species match, bingo! We then map which species were found where. So from a simple sample, we can determine what is present within the broader environment, making the often invisible visible, making the cryptic species easy to find, as we no longer need to see, hear or catch something. eDNA is a valuable tool for those working to protect our ecosystems. We can more easily monitor native species like the popular angling fish, the Murray cod. In the same way, we can monitor threatened species like the dwarf galaxis. We can also detect invasive species like disease-carrying mosquitoes that can enter Australia via our airports and shipping ports. By efficiently and effectively identifying what is present in our environment, eDNA is revolutionising how we manage and protect Australia's fragile biodiversity. If you would like to learn more, please get in contact with us at EnviroDNA. We are a team of passionate eDNA scientists who would love to help.
Okay, so this is being done in multiple different kinds of habitats. I know this video focuses mostly on Australian species, but it can be done anywhere, like the video mentioned, to detect presence, absence of animals, determine if invasive species are present. So it's really pretty neat. I want to do that. I want to do this. There we go. So with this project, the scientists that were involved in collecting the samples, they wanted to look at presence or absence of certain salt marsh mosquitoes within different marsh zones. So they looked at areas near the Tidal Creek, even though I know this is a pond, but Tidal Creek areas and ponds actually as well. Maybe more in like the middle of the marsh, which we'd call like low to mid marsh. And they also looked at the upland marsh edge, where you might see some vegetation changes. You might see some more bushy vegetation like marsh elder or Phragmites, and even around the tree line as well. So they wanted to look at each of these marsh zones to see, where's the most mosquito larvae present. So with doing this research, I thought it would be a really neat opportunity, because eDNA was used in a bulk of analyzing the data. And I thought it'd be really neat to do eDNA activities. And why did I choose eDNA? From this, I could choose, any kind of activity from this whole research project, but I really wanted it to focus on eDNA because it's, based off of real research that we did. It also has really cool real world applications because, a lot of it has to do with management of natural resources. And these are issues that are going on right now, right? So this is a challenge for mosquito control commissions right now. And there's also not too many lessons about eDNA, at least that I know about. If there are any that you know about, please share. I think that would be great. And then it could also be really cross-curricular. You can talk about genetics, you can talk about climate change, human impacts, resource management, all of those things can go into this. So I did 2 modules. One is more of a background in DNA in general, and that's an activity where students extract DNA from a strawberry. It's a really simple activity. You may have even done it before, where you take household ingredients, I think like rubbing alcohol, dish detergent, salt, a strawberry, you smash it all up and you have this solution that is able to extract DNA. So that's kind of cool as like a starting to talk about DNA if you want to do that. But then like the main activity is muddy genes, which is an introduction into eDNA. So how students do this activity is they're broken up into groups and they pretend like they have just taken 3 soil samples from the marsh. And each soil sample is a different marsh zone. And they decode sequences that are in that sample to determine what critters are present in each zone. So it's actually pretty simple and straightforward. So here's a really short demo of it, just to kind of give you a taste of, how to do the activity. So we're going to decode an eDNA sequence from a pretend soil sample. And the eDNA sequences that the students have to decode look like this. They're actually beads on a pipe cleaner. There's 6 beads on each sequence. So there's always these different colors too. I mean, I can use any different color and I'll explain why I chose these particular colors, but there's always going to be blue, green, red, or black. And again, I'll explain why those specific colors were chosen in a second. But so students have about 12 different sequences that represent a different critter. And they pull 1 or 2 or 3 out at a time, however they want to, and they match it to a eDNA probe sheet, or it's basically a decoding sheet, and they match each sequence to the decoding sheet to see what kind of sequences they have in their sample. So for example, the one right here, and students would have to read it this direction from left to right, so starting with blue, ending with black. So out of the 3 critters that you see on the right hand side, which does this match? And you can put it in the chat what you think the match is. Is it the salt marsh mosquito? Is it the snowy egret or salt marsh sparrow? I don't know if this is in the way. I'm going to move it just in case. So what do the beads match with here on the right hand side? Here in the chat. Erin says egret, you are right. Woohoo! Thought I had, there we go. Okay, so yeah, this matches the snowy egret. So if students were doing this in person, they would write on their worksheet that they found 1 sequence for snowy egret. And then they pull out another one, and then they pull out another one. There's actually 12 in each sample. So they actually end up decoding, I think like 36 sequences. So they can do this as a group if they want to split themselves up, or you can do it as a class. But it actually kind of happens pretty quick. And they see how many, what kind of critters are present in the marsh zone near the tidal creek. They see what kind of animals are present near the low or mid marsh area. And then they see what kind of animals are present in the high marsh. So for example, it's unlikely I have in this activity killifish or mummy chug. And so it's unlikely that they will have eDNA for mummy chug up in the upland marsh edge here. Though you never know. I mean, sometimes with eDNA, it's a little tricky because maybe there was a predator and they ate a mummy chug and then the remains are in a tree or something. So you never know. You might actually get fish DNA in a tree, but really unlikely. So it's kind of set up to be, realistic in that, you're going to find maybe more deer DNA sequences up here relative to down here, vice versa with the fish, and so on and so forth. But maybe based on what you already know about mosquitoes in general and your existing knowledge about mosquitoes, where do you think you'll find the most eDNA sequences for mosquitoes? Do you think you'll find it in the upland marsh edge? Do you think you'll find it in the low marsh? Or do you think you'll find it by a pond or tidal creek? If you want to take a guess, go ahead and put it in the chat. So where would you find the most salt marsh mosquito DNA? Take a guess. Okay, it's got some mid-marsh, low marsh, by the creek or pond, red square, mid. Any other guesses? Well, I'm going to be really mean and say you have to do the activity, find out. Haha. All right. So yeah, I have a bunch of different animals incorporated into this activity. And this is where you can find the Muddy Genes full activity sheet in the link I'm about to share with you now. But the activity itself, you only really need a few things, just 3 buckets or containers. I also do this with like baggies, and you put the DNA sequences in the baggie and kids will pull them and then decode, and then they'll go to the next marsh zone, which is the next baggie of sequences. They pull those out, decode. And so the toughest part really is making the DNA sequences out of all those beads and all those pipe cleaners. But I do have pre-made packs that you can borrow from the JC Near. If you're interested, you can just email me and I can send them to you through mail. Just let me know what your mailing address is. And you can do that instead. They're pretty easy to mail out. They're, it's not like a huge box or anything. So if that's something of interest, you can let me know and I can mail them out to you. But I think the toughest part is just getting those together. But if you don't have time, I know you guys are so busy being teachers already, you can borrow one of our packs, as long as you return it, of course, with all of the materials that it came with. But I will show you where you can find this. And I'll put the link in the chat too, as long as with some other links that we talked about. So here's the link, and this is a link to the research project. So it tells you all about what they did during their time from 2016 to 2020. And you can go down here. This is actually really cool. This is a video of the drone in use. So remember I told you about that was one way that they were able to get samples and analyze samples for mosquitoes DNA. So here's a quick video of the drone in use. I'll do that again because it's kind of quick. It's very loud, the drone. And so I just also wanted to mention that there were so many county mosquito control commissions that were involved in this project. So this was like all the way up north, like in the Meadowlands. You can see like the New York City skyline back here. And we also did them like way down in Tuckerton by us, everywhere in between. So, so really huge effort in analyzing samples. Okay, so different things like multimedia, I'll get to that in a second. If you really want to look at the data from this research project, you can. But here are the K through 12 products that I created. So a little bit more about this resource, just kind of gives you a description, basically everything I just talked to you guys about. And then you can go to this button here that says open and download resource. All right, and so here are the modules. This is some background about the project, some biology background for educators. So I'll scroll kind of down here. Also a little bit about past mosquito management, such as grid ditching or open water management. If you want to look into, talking about that with your students, there's information there too. Here's activity 1. that I mentioned with the strawberry. It's called What Does DNA Look Like? And both of these activities I recommend really for grade 6 and up. Here are NGSS standards that it's aligned to. And here are instructions of how to extract the strawberry DNA. I also have what I like to have call a nerd alert in each one of these activities that tie it to real world, what's going on right now and how it relates. So for example, in this DNA extraction experiment, scientists actually do this. They filter and isolate DNA before they analyze it, just like what you're doing with the strawberry activity. Some questions for students at the end that are critical thinking and teacher answer key. And here's Muddy Jeans. So again, 6th grade and up, NGSS standards that it's aligned to, the setup and the prep for the activity, and how to do this. Again, it's really more about the students working in groups to analyze each marsh zone and what eDNA is present in each of those zones. For example, like our snowy egret here. My nerd alert for this is that the bead colors were not chosen at random. They actually are colors of the DNA when they're analyzing it through the machine. So when they are looking at eDNA analysis, they amplify a region of DNA and it glows in a fluorescent color. And it's either black for guanine, green for adenine, red for thymine, and blue for cytosine. So those colors were chosen on purpose because that's how it looks like when it comes up in the machine. And some really, important critical thinking questions as well after they do this. How does this information help county mosquito control professionals, when analyzing this eDNA information? And it's basically like cracking a code. What codes do we find in everyday life elsewhere? And then what are their observations when comparing the DNA from the different marsh zones? Did you notice a particular species was more present in one area than another area? And why do you think that is? So these are really good questions to talk about afterwards. If you do want to put in some time and make your own DNA sequences for the activity, I talk to you guys, or I show you guys how to do that in this section here of how to make them. And then which ones go in each bucket, but I won't look at or go into that too much right now. It's all laid out on the sheet. And then here are the worksheets that the students use to match. So we have different critters like killifish, clapper rail, sparrow, terrapin, crab, deer. And then this is where they record everything when they're doing their analyzing. So marsh A, B, and C. And that is it for the teaching modules. Before we take some questions, I do want to show you one other really super cool thing that I also helped create. I'm not sure if there are any informal teachers here, but 2 things that are kind of cool. Also, this was really more to help the County Mosquito Commission folks when they go and do, outreach events. But if you're one of those people, this might be helpful, or if you're a regular formal teacher, you could use this too. But it's a brochure called Fight the Bite, and it's all about mosquitoes and learning how to protect your family, the kinds of mosquitoes that are around in the areas maybe that you live most commonly. So if you live in a residential area, you will probably see Asian tiger mosquito the most. I know I see that in my backyard like all the time. What kind of mosquitoes you'll see if you're by the coast, if you're, in the forest. And then also mosquitoes and climate change. How does climate change impact mosquito populations? Not just sea level rise, but also intense weather, right? So if we're getting more intense storms, we have more pooling, more puddling, invasive species. And then also with warming of our climate, we could also potentially have habitat range changes. We're seeing that with other animals, such as fish. moving, their typical ranges are shifting, and we are seeing that also with mosquitoes too. So that gives you a little bit of insight into what's going on with that. And then finally, let me go back here. So all of the graphics that you're seeing are available as well. If you have an activity or something you want to do and you're talking about mosquitoes, it doesn't even have to be eDNA. There are these really great graphics that were developed as a result of this project, and they're available to you. They're free if you want to use them in your teaching. So feel free to download these if you'd like. They're available to the public and free and accessible if you want to use them. And the New Jersey Flood Mapper tool, that's how I got my maps. If you want to look more into that and kind of dabble with Flood Mapper, you can access Flood Mapper this way too, though I think someone also put it in the chat, which is great, so thank you. And the data, if you're interested in really looking at the data that was collected, I believe you can access that as well. You just have to request that. So, but you can click here to learn more about how to do that. So I think that is it for me and the modules and other outreach and education materials that I developed for this project. I will go ahead and, whoop, hold on, let me go back. There we go. I will take any questions, or if you guys want to discuss ways of extending or improving the materials that I made, I'm all ears. I have done this activity with teachers. I have not done them with students yet, so it would be, because of the pandemic mostly, it would be really neat to get feedback. If anyone's interested in providing me feedback, I will definitely be happy to chat and collaborate if you're interested. So, and again, email me if you're interested in getting a mailed copy of all the materials as well. And that's my email there. So any questions?
Caitlin, looks like someone's asking what grade the activity is geared towards. I know you said you just did it with teachers so far, but what grades are you shooting for with this?
6 and up.
Grade 6 and up?
Yes.
Yes. Perfect.
For both of them. I think maybe younger students could definitely do like the matching part if they have an older student present with them. But mostly the whole activity is really for 6 and up.
All right.
Oh, we have an informal educator too. Cool. Could be great for parks. Yes, absolutely. And you can download that brochure again through the website, which I believe I put in the chat, which is the NEARS. Yes, I did. So that I'll put it in once again. It's the NEARS Collab Lathrop. This is where you can get everything: Muddy Jeans, the outreach materials, the drone videos, all this stuff. Can the brochure be shared on my town's newsletter? I don't see why not. I mean, it's open to the public. So I don't see why that would be a problem. Yeah, again, we made them mostly for the county mosquito folks and these modules as well, because they were requesting it, but it's now open to anybody who wants to use them. They're public material, so.
I have
Uh-huh.
one possible comment, if I may.
Yeah, go ahead.
A possible suggestion for the beads, because my first thought when I saw the bead colors, I mean, immediately I knew what they were for, but there could be kids with color blindness. And if you can get shaped beads,
yeah,
you can still go with the colors, but change the shapes of the beads so that it has the patterns as well.
Yes, I have actually thought about this as well. When I did this, the color beads that I got were like the green and the blue were very similar as well. So I like that idea of doing different shapes of beads or beans. Even I've thought of using, maybe that can have like different texture. But yes, that's a great alternative. Thank you. Can you guys, the educators that are on today, do you feel like you could see yourself doing this kind of activity with your students, or is it sort of not really in the realm or subject area? Or if you want to put any insight into the chat, that would be helpful too, for me to know as well if I need to make any other adjustments or do something different for next time. I'll stop sharing too.
Another interesting note, I worked for Department of Ag, and they did use eDNA to study where spotted lanternfly can be found.
Oh, great. Yeah. That's very important right now, very relevant for sure. Yeah.
All right. Someone's asking, how affordable would it be to participate? What kind of equipment would you need? I'm assuming that they mean in the eDNA study.
Oh, so I know there are eDNA like kits that you can get. We don't have those available. We just do the activity that sort of simulates what it would be like if you were using an eDNA, instrument. But there are equipment that's actually portable that you can take out into the field if you really did want to use equipment and analyze eDNA. I actually have looked into this myself, and I think there's some equipment out there for maybe like $500 to $600, I would say, that actually could be used with students. But I don't know the brand name of it off the top of my head, but they do exist for, for scientists, but also for students to just go out, if you want to go out in, the schoolyard even and take some samples. Maybe that's something I can look into if you'd like to touch base again. I do have that information. I just don't know off the top of my head. All right, perfect. Sounds good. People say they'll use a strawberry lab activity. Yeah, and there's also, I believe, even YouTube videos of how people have done the strawberry lab. So that's something, even if you Googled it or went onto YouTube and looked it up, there's lots of variations of the strawberry activity, and it's very easy. I feel like you could do that with maybe some younger students. All right, well, anything else? And I think this is kind of neat, especially it includes genetics and something that's, includes lots of kids' favorite critters. So if you have any other, materials or suggestions to expand on this, let me know. You can email me as well.
Well, if there's no more questions, which, last chance, if anyone has more questions, you're certainly welcome to. We have a few more minutes. But I really want to thank you, Caitlin, because it was a really great presentation. It looks like you're ready to bring this to schools. Do you get to teach too?
I do, yes. Just not lately because, of COVID. We have pretty strict regulations on, doing in-person programs right now. But I definitely, for any South Jersey schools, I can do this with your class as well, as long as I'm able to, get the green light to do those programs again. Yep.
Nice. Nice. Well, you did a great job today. We appreciate your time, and we're really thankful that you've come, and we thank you for all the wonderful work that you're doing as well. I just want to do a few reminders before we end the session today. We're going to be posting an evaluation link. Thank you, Erin. Please fill out your survey before you Before you forget what you've done today, let's fill out those surveys at the end of each experience. Check out the in-person field experience. Even if you signed up for the conference already, it's not too late. You have another 40 minutes to sign up for any of the in-person field experiences up until 4 o'clock today. Thank you to our conference sponsors again, PSE&G, New Jersey Natural Gas, and our anonymous donor, and to BASF and Grounds for Sculpture for supporting our field experiences. Thank you. Thank you very much. Great job. Really great job today.